Hi, and welcome to the Unserious Science Podcast.
Episode 1. So we decided to make this podcast to celebrate and uplift LGBT people in STEM. And what we are trying to do is get people on to talk about their favourites or theories, pet peeves, and white lies that may be presented in popular media about their profession. So we're starting today with one of our lovely co-hosts.
Hello.
I'm the other lovely one, obviously. So I'll just introduce myself and then hand over to Bex. So I'm Dr. Eleanor Townsend. I'm a microbiologist.
Why did you say that with so much flavour?
It's a spicy profession.
Okay, great.
I'm a microbiologist and a chaos bisexual. There we go.
Is that how we're officially...
That's enough about me.
Okay, perfect. I am Dr. Rebecca Smith, or Bex. I am a statistical geneticist, but please don't let that put you off. I am also a chaos bisexual, but I'm also a romantic.
Lovely. Jubbly.
I feel like we should start this again.
I thought that was fine.
No, I think we're good.
Okay. We'll cut out the lovely jubbly. That was weird.
I'm leaving it all in. I'm editing this, so this is all staying in.
Okay. So, what are we talking about today, Bex?
So, yes, Eleanor, as you've just said, we are co-hosts of this show, and our aim in the wider scope is to get guests on to talk about different areas. Obviously, we're both biologists, so we feel it'd be quite limiting if we were just talking about biology. But because it's our project and we want to show willing, we are going to be presenting our topics first. So today we're going to be talking about Jurassic Park. So Eleanor, have you seen Jurassic Park?
I have. I feel like I saw half of it in the 90s as a kid. Was it out in the 90s? Yeah, it was out. 93. I was 1 year old when this came out.
Stop.
So I saw some of it when I was a kid, and then I've seen it the whole way through as kind of a young adult.
Okay.
Yeah.
But you're aware of the basic premise?
Yeah, I think I actually saw Jurassic Park 2 before I saw Jurassic Park.
Oh, you poor thing.
Okay.
It's not as good as the first one.
Yeah, and then I've seen like the more modern ones.
Oh, the Jurassic Worlds. Yeah, we're not going to do talking about those. I refuse.
Okay. I know just the first one. I think I went to the cinema. Don't know. But I know the idea.
Anyway, we are going to be talking about the first Jurassic Park movie. So if you haven't seen it, and if you haven't seen it, what are you doing here? Go watch Jurassic Park. But it is a movie from 1993 based on a book by Michael Crichton in 1990. And the basic premise of the movie is that they're making a essentially safari/zoo for dinosaurs. And it's made by this wealthy guy called Dr. Richard Hammond. Dr. John Hammond. It's made by this rich guy called
Because we were talking about Brainiac
earlier.
It
was. It's made by this rich guy called Dr. John Hammond, who's played by Richard Attenborough. And it's got some amazing actors in it, such as Laura Dern, who I'm in love with, Sam Neill, Jeff Goldblum, etc., etc., etc. But the basic premise is they make a Jurassic Park for dinosaurs, and spoilers, it goes very wrong. And people are injured/die. So not an uplifting film about the future of cloning dinosaurs. But the thing I really want to talk about is about the genetics underlying Jurassic Park. So do you have a background in genetics, Eleanor?
I'm a wee dab hand at the old molecular biology, but I wouldn't really describe myself as a geneticist. And not of the eukaryote kind anyway, but we'll give it a bash, eh?
Okay. As you may expect from a film made in 1993 and a book written in 1990, the genetics is not amazing. Obviously, this film and book came out 21 years and 18 years respectively.
Yeah.
Well, the book came out in 1990, so 21 years before the first draft of the Human Genome Project was published, and the movie came out 18 years before that. So obviously a big gap between the science fiction and the knowledge that we later had of genetics. And obviously there's been a lot of sequencing and investigation in genetics before the Human Genome Project. I know that before the Human Genome was sequenced, or at least the first full draft of the Human Genome, we had a rough draft of like the mouse parts of the mouse genome.
Yeah.
We had, I think, Drosophila, so like fruit flies.
Mm-hmm.
I think we had yeast.
Yeah. Saccharomyces.
Saccharomyces, yeah.
Surely like a C. elegans as well, the little wormy things.
A lot of sort of like simple multicellular
Models,
as well as
yeah.
single cellular. So like bacteria and things like that. Things that have a much, not necessarily, a smaller genome, but a lot of things that had a bigger genome as well. So the technology of sequencing at the time was very labor intensive. The Human Genome Project was made via Sanger sequencing, which is extremely short read, 50 base pair to 150 base pair sequences. So the human genome is 3 billion base pairs.
I can't do the maths.
No, I don't want to do the maths. But essentially, there was an awful lot of sequencing and tiling, which we'll come on to a little bit later. Okay. But essentially, just trying to like match up all these bits and pieces. Anyway, we're getting slightly off topic.
Okay.
But
whoops.
That is fine. It was going to happen. So basically, before we had the first draft of the human genome, we had these other organisms' genomes, and they were incomplete in a way. Like there was a lot of repetitive sequences and all genomes, which are generally quite hard to sequence, especially in Sanger sequencing. Yeah. So they tend to get omitted or not fully done just because sequencing slips a little bit and you lose those repetitive sequences. Yeah. But let's start with dinosaur DNA. Okay. In terms of how this works in the movie. Just to remind you, Eleanor, they get DNA from blood-sucking insects that were around at the time. And then they, I assume, or they assume, have bitten dinosaurs.
Okay, assumption number one.
Assumption number one, these bugs have bitten dinosaurs, apparently. Okay. Then they extract that blood, extract the DNA, and then use that to clone dinosaurs. Yeah. So obviously they don't know what they're getting until they do it, I guess.
Yeah, it is a bit of hope and a prayer, isn't it?
Yeah. Could be anything.
Were the bugs in amber as well, as I remember that right?
I think yes. It's tree sap.
Yeah, that's yeah.
So they extract the bugs, they extract the blood, they take that DNA from the blood, and then they use it to make dinosaurs.
Could be anything. They're hoping it's a dinosaur.
Yeah, it's a wing and a prayer. But you know, hopefully, if there were lots of bug-sucking insects around at that time, which there should be a lot of samples. And if you can find, I don't know where they're finding all these bugs, but essentially, if they can find enough, they can definitely get a good variety of DNA.
Okay, yeah.
The problem with this, and as someone who has a passing knowledge of genetics maybe, DNA isn't very stable, especially over a long period of time. So when we take DNA from very old samples, so 1 million years, let's say, for example, a lot of it's going to be broken / missing / unusable. But when you take samples from things which are tens of millions of years old, yes, you can imagine what's going to happen. It's basically going to be mostly gone.
Tiny fragments.
Yeah, tiny fragments. So there's been a lot of attempts to try and extract ancient DNA, as they call it. And basically, over millions of years, that DNA is going to start breaking down. The integrity is going to be lost.
Yeah. Especially, I'm imagining now, my Jurassic Park knowledge is low. Okay. But I'm imagining this is quite a tropical place, so quite like low, high humidity, high temperatures.
I don't think all the samples came from the location that they're making the park.
Okay.
I think that they made them because you find dinosaur skeletons all over the world. Yeah. Like America and Europe and Asia. I was trying to think of continents.
Another place apart from America.
Yes, we are totally not American. I don't think that's going to be super necessarily a problem. Although samples that do come from those places obviously gonna have more an issue. But just in terms of research into ancient DNA, we have found DNA that's about 2 million years old that we've managed to find and investigate, but it hasn't really been able to be sequenced. So we don't really know the sequence of it. And from ancient DNA, the fragments are going to be very, very small. So like 40 to 500 base pairs is roughly what's been found currently. But the problem with this generally is that and this is based on a study of New Zealand moa, which are a bird. And they found DNA which is certain years old, and then they sequenced it and seen how much of it is degraded. And they determined that the half-life of DNA is 521 years.
Okay, specific.
Very specific, which means that every 1,000 years, you're going to lose 75% of the DNA.
Yes, yeah.
500 years, you lose half.
And then another half, which is actually a quarter.
Exactly. So this keeps going. And if we're looking at things which are over 1 million years old,
that's small.
basically it means that we're likely to have less than 1% of the DNA remaining.
Okay, yeah.
Which is not ideal.
I suppose it depends how much you start off with, right?
Well, if you think of like well, it also depends on how it's fragmented, but if you think about the human genomes, if we think the 3 billion bases and we fast forward in time and someone finds a sample of human DNA millions of years in the future, we would have very little.
Yeah, and I suppose the other thing is that it doesn't degrade at like a constant. So some parts would degrade quicker than other parts.
Yes.
Because of the...
Well, it would be somewhat random as well as to what was maintained. Obviously, it depends on how DNA is organized. And again, we'll come on to that a little bit later.
Okay.
But obviously, DNA is normally arranged into chromosomes, and chromosomes have structural proteins which keep them stable.
Yes.
But through the action of bacteria and just general environment and like our own cellular machinery of like DNA repair, those obviously break down. And so essentially, the DNA would just become randomly fragmented and not really usable.
Yeah. Bit of a difficult puzzle to put back together.
I think it's like making a puzzle where you have one piece and all the rest is missing.
And not entirely sure what the picture is on the box either in Jurassic Park, because
No.
they don't know what dinosaur they've got.
Well, but also we don't have a full sequence, so it's not even like we can align it to anything.
Yeah, so you got no picture,
No
guide,
No.
no instruction
puzzle
book,
piece.
one puzzle piece. Good luck.
Yeah. I mean,
Have
it'd be
fun.
interesting. It'd drive me mad. Yeah.
I hope they paid those scientists a lot in this film.
Oh, yeah. He was like a multi-millionaire, billionaire. I don't know what the transition from the 90s to now would be, but I assume very wealthy.
Okay.
So I'm sure they're paid a lot. And also he's expecting to make some money back from dinosaurs.
Pay your workers. That's what I say, Dr. Hammond. Make sure you pay those poor kids in the lab.
Those poor kids.
Yeah, there's little PhD students busting their ass to
Can
get
you
a
imagine doing a
Triceratops.
PhD in...
Jurassic Park.
Yeah, that'd be awesome. What's your PhD in? Oh, I'm cloning dinosaurs.
Yeah.
You'd be the coolest PhD
Good
student ever.
luck making it to your Viva though, guys. I saw the bit with the raptors.
Yeah.
Yeah. Okay.
The genetics.
Back on topic.
Back on topic.
Back on topic.
So that is obviously main problem number one, is that
Okay.
I think that it would be, or say scientifically, it would be very difficult or almost impossible. It would be impossible to get a full genome
Yes.
of a dinosaur. And then if you are then using lots and lots of dinosaur specimens from the same species, being able to tile that together, like so these small fragments, building them on top of each other, being able to get a full picture,
Yeah.
especially because certain reptilian species, birds, these are all dinosaur descendants. So you crocodiles. You could use some of these as a sort of like
Template.
yeah,
Inspiration.
exactly.
The picture on your box of the jigsaw puzzle.
I get you. But I mean, in terms of, it would be a slightly wrong picture depending on what you're starting.
Yeah, I
picture
guess.
with.
Yeah, because there's this whole thing about them having feathers or not now, right?
I mean, it has been. I think everyone agrees that dinosaurs had feathers.
Okay, but Jurassic Park didn't have feathers.
Again, we're not getting into like
Okay.
because like there's a lot. I really like dinosaurs, obviously. So I've been doing a lot of just interesting reading about dinosaurs, and I think that most people agree that they mainly evolved into birds. Birds are their Birds are their descendants.
Yeah.
But they likely had feathers based on fossil records and things like that. So, which is kind of cool. They're much more colourful than Jurassic Park painted them.
There we go.
I know, I like birds as well. So.
Flamboyant dinosaurs.
That would be awesome, wouldn't it?
It would.
It's like full on peacocking.
Yeah. I don't know.
I think it'd be great.
I quite like parakeets and stuff. So
Yeah, that'd
I'd
be nice.
enjoy it. Do you remember there's one dinosaur that kind of goes
Yes.
and like sprays like acidy stuff? Am I
Yeah, that's
remembering
not
that?
real either.
No, I know, but imagine if it was that, but with feathers and like
Frills and feathers.
and Elton John glasses.
Where would they get them?
I don't know. I just want this like flamboyant sort of
A jaunty hat.
carnival. Yeah, just put some glitter on him.
I mean, you could, but you'd likely die.
Worth it.
Worth it.
That's what my PhD would have been in.
Death by fabulousness.
Yeah, just can you put glitter on a dinosaur? First publication. No.
I mean,
Get it in
technically
nature.
yes,
Get it
but
in nature,
also no. Get it in nature.
Get it in nature. No.
I want to
The
see
figures
that
would be
fall.
great. The figures would be so good.
my
The writing, not so much.
I want to see that fall across an editor's desk and see what their first reaction would be. I don't know. I've
not
never
gonna
edited a
happen.
journal, so
They're not. Yeah, it's not fun.
other issues with this. So the idea of sequencing very old DNA is obviously something that is a problem, especially if by the time we get the sample, 99.99% of it has been degraded, and it's not because it's been stored God knows how. It's especially for the ones which are preserved in amber/plant sap, it's not going to be temperature controlled. It's going to be open to fragmentation, and if you've ever done any genetics work, you'll know that how you store your samples is very important, because they even very short amount of time can break down if they're incorrectly stored, if you just leave them at room temperature or whatever.
Yeah.
So one thing which could be a way to get around this is like using samples in permafrost, right? So if we
Nature's freezer.
nature's very cold place. So if we manage to find good specimens in frozen areas, into permafrost, then maybe that DNA would be better. No, it'd still be really badly fragmented. This is like, so I had this same thought where I was thinking, oh, maybe like freezing if they're really low temperature and maintained at that. Similarly, it would still be extremely badly fragmented.
Because did they not do something with like a mammoth?
Yeah, so there was a 2 million year old mammoth specimen, something like that, which was in permafrost, and they managed to get, it was highly degraded, and they did manage to get a lot of the genome.
Okay.
But still, it was like 70-ish%, I think, of the genome. But again, it's not the worst thing, right?
Yeah.
But again, it's the difference between 2 million years and 66 million years.
Yeah, that was going to be my next question.
Yeah.
Yeah.
And also, we have elephants.
Yeah, that I was also going to say that. You've got a picture on the front of the puzzle box.
Yeah, exactly. We know that mammoths are very similar to elephants.
Yeah.
So we know.
So you can make assumptions. You could say, oh, I'm pretty sure like this bit would be the same as an elephant.
Exactly. It's unlikely that there's been a huge amount of change in chromosomal rearrangement and stuff, especially for species which are very similar to each other, or historically supposed to be similar.
Still an assumption though.
It's a very big assumption. But yeah, so permafrost could be, but again, still we'd be losing an awful lot of DNA. There's way more we could talk about in terms of DNA fragmentation, super interesting stuff, and about how the samples would likely be incomplete and how we don't have a good template necessarily, depending on the species. Obviously, if we have the more the later dinosaurs, which are more avian-like, so more like birds, we obviously can make some assumptions about bird DNA, right? We have a lot of bird DNA.
Yes.
So there is potentially templates we can use. So chickens, for example, quite close to avian.
I like chickens.
Yeah, I like chickens too. They're very close to avian dinosaur species.
Yes.
And
you can see it sometimes.
You can. Well,
yeah.
They actually used chickens to model the movement of the velociraptors in Jurassic Park.
Okay, maybe that's tainted. Maybe I'm thinking, oh, dinosaurs act like chickens, but it's really all the films I've seen. the dinosaurs are acting like chickens, but like the chickens aren't acting like dinosaurs. You know what I mean?
No, I know what you mean. Like, has the model tainted?
Shaped my idea. Yeah.
No, I agree. Like, the things we don't have necessarily, like, apart from like sharks and crocodiles, we don't really have living dinosaurs. So what do we base that behavior on? So the last thing I want to talk about in terms of DNA, in terms of the samples that could be used. So let's ignore the fact that DNA would be highly degraded, fragmented, some might say unusable.
Yes.
Let's ignore the fact, and we haven't really spoken about this, of them using frog DNA.
Yeah. We'll ignore it, eh?
We'll ignore that because there's just so much. Like, we all know that birds are more closely related to dinosaurs than frogs are. Filling in the fragments, the missing parts with frog. One, your dinosaur would be 99% frog, so not really a dinosaur. Two, the genomes of frogs and birds / dinosaurs wouldn't really be able to slice together in that way, especially with so much being coming from frogs. And three, the sex change stuff, again, that is something that does happen in frogs. Like, some frogs become either hermaphroditic or change sex. But so do birds.
Okay.
So, chicken story. A very anecdotal chicken story. When my mum had a lot of chickens, we had one called Bernie, who we had hatched. So she changed sex. So basically what happens in chickens is that they're hatched, I say born, they're hatched with 2 ovaries, but only one develops.
Okay.
So, and again, things I've learned about chickens from doing this podcast. But essentially, yeah, so they hatch, while they develop, they have 2 ovaries, and when they're hatched and they reach maturity, they only have 1.
Okay.
Which means that, obviously, if that ovary is damaged in some way, all their hormones go into imbalance.
Yeah.
So what happened with Bernie, and what's happened with a lot of chickens I've seen online, and like I've been looking it up online to sort of like confirm it wasn't just a crazy dream I had, but essentially Bernie, she basically grew a massive comb.
Yeah.
She grew like a big wattle things.
Yeah.
She started trying to crow.
Aww.
So she became a boy chicken. Obviously infertile, and again that would be the issue with like the bird becoming changing sex. Is that they likely wouldn't be able to breed by themselves.
Okay.
Yeah, it does happen.
Yeah.
Which is kind of interesting.
Yeah.
I don't know if it's specifically chickens. I assume other birds do as well. But.
Yeah, I'm not sure. But there we go.
Yeah. Anyway, that's a story. A story that's somewhat related.
Bernie the chicken.
Bernie, R.I.P. Bernie. She was a crazy chicken. Okay, so going back, ignoring frogs, ignoring DNA degradation, ignoring all that issues that I've spoken about before. Let's say we do manage to get a DNA sample that is usable, and we manage to tile it onto something, we manage to get a full sample. Another problem comes from contamination.
Yes.
So what do you imagine, Eleanor, might be the problem with contamination of DNA samples?
Well, if you're working from something that you're not 100% sure what it is, inside a mosquito type creature, inside tree sap. I'm not sure if tree sap contains tree DNA. Maybe it does.
I don't actually know.
I don't know. No, I don't think it does actually.
No,
but never
it's
mind.
product,
Anyway,
isn't it?
so you've still got like mosquitoes. So if you've got your sample, you've somehow got to get all the blood, which is dinosaur, even if it's not degraded. And you've got to get it all out without getting any mosquito in there, because otherwise you're going to have a like mush of mosquito and dinosaur. And then now, this I know from micro, when you're working with something that's very small, you try and amplify it up, any problems or contamination are going to get amplified up as well. So if you start off and you've got like one tiny bit of mosquito in there, but also one tiny bit of dinosaur, and then you're like, make it bigger, make it bigger, make it bigger. But then it's still, you're making the bits that you don't want bigger as well. So you're just going to have big old mush of mosquito and dinosaur. And that's accounting for it all staying encased in tree sap, and like no one sneezes on it.
The lab tech.
Yeah, which people do. It's
100%.
very easy to contaminate stuff, isn't it? Like this is this way you see forensic people in all of the kit.
Yeah.
So yeah.
But even so, yeah, you're 100% correct. Like the likelihood of having like mosquito, whatever else in there, but also, you know, bacteria.
Oh yeah, forgot about them.
That's
my job. That's my job. Bacteriologist.
Viruses.
Oh, yeah, them.
Like
yes.
anything else that's in the environment.
Yeah. Okay. Yeah.
And again, if we don't know what the starting template is.
Yeah, they do operate on. No, I take your point. I'll allow it.
I mean, it's not the main point I'm making, but it's just something to think about as well.
They operate on slightly different systems, bacteria and eukaryotes.
Yeah.
So you might be able to shift those out. Viruses is a whole another thing, which we won't get into.
Or like fungi.
Fungi, eukaryotic, so it would be a little bit more difficult to shift those out.
I'm just saying, there's a lot of other genetic material in there.
Just getting in, just a mush.
Yeah, a mush. A mishmash of mush.
A mush.
So that's DNA.
Yes.
Okay, so that's like just the molecule of DNA that I know and love.
Okay.
And again, there's even more I can say about this in terms of like the organization of DNA in dinosaurs. We don't 100% know what the organization would be. Obviously, in humans and most creatures, we have chromosomes.
Eukaryotic creatures.
Eukaryotic organisms
yes.
have chromosomes. Again, this is about like how DNA is organized and structurally made around
protected.
Stability, right? That's the whole thing. But what I again found out when I was researching this is that if dinosaurs are more closely related to birds, and even if they are related to frogs, which they're not, and lizards, lots of these creatures have microchromosomes.
Okay.
So humans, we have 23 pairs.
Yes.
Of chromosomes, which are arranged into, 1 through 22 plus our sex chromosomes. Mm-hmm. Birds, chickens, I've got a lovely picture that we can put on the Instagram as well, have 39 pairs of chromosomes.
Okay.
Most of these being really small, like...
Yeah.
As you're seeing in the picture.
Yeah, I'm just looking at it and they just get smaller and smaller.
Yeah. So chromosomes are arranged by size, so the largest one will always be 1.
Yes.
But yeah, number 38 is absolutely tiny.
How many genes are we looking at?
Still quite a few.
Okay.
I did look it up. It's like dozens of genes.
Dozens.
Not like a huge amount, but dozens.
Yeah.
So there are still quite a lot of genes on those chromosomes. But yeah, I just thought it was really interesting.
I've just clicked on as well. That's like 38 pairs.
Yeah. So
and we've got
76, or actually 78 because of the sex chromosomes as well.
Yes.
It's a lot of chromosomes.
There's a lot of chromosomes. Fair play, chickens.
And a lot of birds and frogs
all of the birds.
reptiles.
Yeah. Good for you. Good for you. All your chromosomes.
The structure of the chicken genome is just fascinating. You have to do more reading on it.
Yeah.
I never really thought about it. I don't even know what like the function of these microchromosomes are. So the definition of a microchromosome is about one. So from my notes here, they have chromosomes ranging in size between 250 megabases.
It's big.
It's pretty big, and 3.5 megabases. So quite a big difference. But it only has a genome of 1.1 billion gigabases.
Okay.
So it's wild. Anyway, I just thought it was interesting.
It is interesting.
So they have quite a big variation between the chromosomes, and even their biggest chromosome is quite small.
Okay, so just a bit more spread out.
Yeah, like much more varied.
I've never really thought about you hear about animals having different numbers of chromosomes compared to us because...
there's lots of stuff to say about that.
Yeah, like crossbreeding with like ligers and mules and all of this. That's the only time I've really thought about it, but I haven't really thought about the size of the chromosomes as well.
Last thing I want to talk about now
is
cloning.
Okay.
So the entire crux, after we get over the DNA, after we get all over this stuff, just imagine we have a good sample of DNA.
Yeah. And a good template.
And
no contamination.
No contamination. We have to fill in the gaps if there are any.
Okay.
It's completely pure. We know it's a T-Rex, whatever, right? We know we want to clone this dinosaur because we have to do it because we're starting with just DNA. We can't just use DNA to make a creature. We have to go through development, things like that, right? So that's where the cloning comes in. I'm going to give you a list of creatures which we have cloned successfully.
Yes. Number 1, Dolly the sheep.
Number 1, Dolly the sheep. Yes.
Sinner, taxidermied. R.I.P. Dolly.
So I'm going to give you a list of creatures which have been cloned.
Okay.
And I want you to tell me what you notice about them.
Okay.
Okay? It's a
Sounds
quiz.
like a
And you can do this at home.
Oh, God.
So when you think you've got an answer, lock it in and then I'll tell you. So yes, Dolly, sheep.
Hi.
Ferrets.
Yeah, love ferrets.
Yeah. Wolves.
Okay.
Cats, which is my favourite cloning story. We might do that another time.
Okay.
Cows.
Okay.
Rats.
Yep.
Camels.
Okay.
Carp.
Okay.
Frogs.
Okay.
Fruit flies.
Okay.
Macaques.
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 What's a macaque? A monkey?
Yeah.
Okay.
So there's two types of macaques.
Oh, yeah. Yeah, with like a QUE.
Yeah. So that's a small sample of all the creatures that have been done. There's like a huge long list of what do you notice about these creatures?
Wow. I was like, all mammals. And then you whipped out carp. And I was like, ruined.
Yeah, sorry, broke that theory.
Yeah. All fairly domesticated? I'm not sure. I'm sorry.
No, that's fine.
I was so on the mammals track, and then carp just came in and slapped me around the face and ruined it.
So, that's fine. None of these lay shelled eggs.
They don't.
They don't.
So I wasn't too far off on the mammals.
No, you weren't too far off
thinking,
until the carp, frog,
So
and
the
fruit fly came off. Also, none of them have large eggs.
No, I suppose the ones that do have eggs are kind of small and squishy.
Exactly. So, cloning has been around for a lot longer than people realise. It started kind of in the 1950s from embryo cloning. So essentially that's when you fertilise an embryo and then you basically remove some cells. And because it's early on in development, those cells will just replicate again.
Okay, so kind of like identical twins.
Yeah, exactly. So it's essentially taking a very early embryo, splitting the cells, and then those cells continue to replicate so that causes multiple offspring, which are genetically identical. But most cloning nowadays, especially, or in terms of how we learnt more about cloning, is from adult cells.
Okay.
Yeah. So, for example, Dolly, that's been around since like the mid-90s. So still very early on, about the time Jurassic Park came out. But yeah, so Dolly was made from a mammary cell.
Okay.
So what we do in all these scenarios is we take genetic material from either an embryo or from a mammalian cell. We get an egg. We remove the original genetic material and replace it with the DNA we want to clone.
Okay, so sorry, when you say egg, you mean kind of like ovum?
Yes.
Cell?
Yeah.
Not like a hard-boiled
Well, well, no, not a hard-boiled. They're all eggs, right? Like all things start from eggs.
I'm just okay. I have a question. I'm just thinking. So you've got you have your like little sheepy embryo or whatever, and presumably they put it inside another sheep to sort of gestate.
Yeah.
Brew.
Yeah.
Pop out the cauldron.
Witchcraft.
It's got to marinate in it.
This is disgusting.
But what would you put a potential dinosaur? Where would you gestate that? Is that the point you're coming to?
That's one of the points I'm coming to.
Okay, because like when you have crossbreeds of dogs and you have like a Great Dane crossed with a Dachshund. If the mum's the Dachshund, she dies. Yeah. Because those puppies are too big and they explode her.
Sorry, that's not funny, but the way you said it and looked at me was awful. Yeah. You're 100% right.
Yeah, this is where I was going. So the gestation of these sort of wee sows, because I mean, with a hard shelled egg, you can pop them in an incubator.
Yes.
I don't know if they maybe do something inside first.
Well, there's a couple of theories as to how we could and why we can't clone currently, at least, and this might change as of 2025. We can't use these like large shelled eggs. So yes, you're right. With things like Dolly, with all these cloned mammals and things like that, the egg is accessible in terms of we can extract it, remove the genetic material, replace the genetic material, and put it back into a uterus or into, like, if it's a soft, small egg, then we can do that.
Yeah.
But the problem with birds and dinosaurs...
And some reptiles.
Although reptiles do have small eggs, but they are quite large. But dinosaur ones, I think, are hard. I can't remember.
Okay.
They're fossilized, the ones we find.
Yeah,
so I don't know. So the problem with using these kinds of eggs is that if you were going to do it in the same way of you would for a mammal, you would have to be able to get at the egg prior to the shell being placed.
Oh, okay.
So you'd have to be able to get into the ovum and then put it back in. Okay. Well, not even the ovum, because the actual fertilization happens outside the ovum. So essentially, not the ovary. So essentially, once the cell is released from the ovary, then it's fertilized, then the shell and everything is put on and it's laid.
Yeah.
But being able to do that in chicken is quite hard. You have to be able to time it perfectly to be able to put the sample in the right place and then replace it in the chicken before the shell and the yolk is added, blah, 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 blah,
Oh,
blah.
geez. Okay.
Or whatever creature it is you're using.
Yeah.
That's an egg, a shelled egg.
Okay.
The other issue is, and you might be thinking, why can't we just take a chicken egg and cut off the top, which we can do. We can 100% open and put plastic on top. Have you seen that?
Yeah, but they don't. It's a bit risky that they don't always work.
Well, when I was an RA, I did work in a lab where they were literally. It's the first time I'd ever seen it. They would take fertilized chicken eggs, cut off the top, cover them in plastic, and electroporate DNA into them to cause genetic mutation and things like that. But the problem with large eggs is that they are large.
Okay.
So a main issue with this is we can't put a chicken egg, I'm just using chickens as an example, but we can't put a chicken egg or any kind of bird egg under a microscope.
No, because it's too big. It doesn't fit. Because
it's way too big. It wouldn't fit. And also being able to find the genetic material inside a massive space of an egg, which is absolutely microscopic sample.
Yeah, okay. I'm just the headache of this. I can't get. Yeah. And all dinosaurs lay eggs.
Yes.
Okay. This business plan for Jurassic Park.
Yeah. So even if we're looking at something like a mosasaur, which is like a shark-like dinosaur, even if they laid shark eggs, so soft-shelled, like mermaid's purse kind of style eggs, like stingrays and things. Have you ever seen those?
No, but carry on.
We'll have to look those up later.
Okay.
But yeah, so they have those sort of horned eggs, square eggs that are soft. So even if they laid eggs like that, or reptile eggs which were soft shelled, being able to find the genetic material and replace the genetic material would be extremely difficult, if not impossible.
But also because if we're cloning a dinosaur from ancient DNA, but we haven't got a dinosaur egg to put it in anyway,
you'd have to go for something that's close.
Yeah, so even an ostrich egg is massive, but trying to get a baby dinosaur in an ostrich egg, it would burst out the shell before it was done.
Well, I guess you could start with a small, a microraptor or something. They're about the size of
and then use the microraptors.
No. I'm just mean as a proof of concept, I guess.
Okay.
You could start with a small,
but
or one of the avian dino species which are closely related.
Yeah, it's not very Jurassic Park. You want your T-Rex, don't you? You want your Diplodocus.
If you started with a T-Rex, that's crazy. Surely you'd start with something new and work backwards, as opposed to being I'm just going to go for T-Rexes. Obviously, that is a problem for cloning. But again, while I was doing research for this, I came across a really cool study, and it is related, about making chimera animals.
Okay, back to ligers.
Not really. A liger is a crossbreed.
Yeah, cross species. Yeah.
Yeah, but a chimera is an animal or plant that has a mix of genomes in it.
Oh, okay. So like those tomatoes that have got a fish gene in.
Kind of.
Stop them from freezing.
Well, no, actually, it would be some cells are like a chicken and some cells are a dinosaur.
How does that work?
Exactly, right? So this is true amazing thing. So there is obviously a lot of work being done to try and bring certain creatures back or save them from extinction, right?
Thylacines.
So there's a lot of work being done to try and protect and up the number of creatures of a certain species which are currently under threat of in danger, as well as to bring certain species back which may have. So the mammoth is one of those, right? They're thinking about, I don't know if I believe that it's ever going to happen, but, or if that it's ethically okay to happen, but it is something that people are trying to do. So one way they're trying to do this, especially for things we don't have a lot of animals for breeding, is creating chimeras.
Okay.
Where essentially they put cells in another creature, and then some of those creatures will become, when crossbred, like when bred, the species you're hoping for.
Okay, so do you take like some cells and some cells like very early in development?
So the example I saw, they cloned or made a chimera of chickens and bustards, specifically.
What did you just call me?
Specifically, Hubara bustards, which are a like mostly ground-dwelling bird, which is not too far from a chicken, to be honest, omnivory, found in Africa, which is endangered.
Yeah.
Or certain species of it endangered. What they did is they took gonad cells, so like testes, cells of these bustards, and they basically put them with a developing embryo of a chicken.
Yeah.
So essentially, this ended up with a mixed gonad of chicken and bustard, meaning that when they were creating sperm, some of them would be chicken and some of them would be bustard sperm.
Okay.
Then they bred these chickens artificially by impregnating, that's not right for chickens, by fertilizing bustard with the sperm from these chimeric chickens.
Okay,
these
and then
roosters.
Half of them would have.
Well, it depends on the proportion of cells that were in there. How
but so some of the babies would be full chicken.
No. They bred them with female bustards.
So some of them would be half chicken, and some of them would be full bustards.
I don't think the half chickens would actually fertilize.
Oh, they wouldn't be viable.
Yeah, but some did fertilize.
Okay.
As bustards.
There we go.
Yeah.
Good for
Only
them.
one survived.
Oh.
Out of quite a lot of work.
Okay.
And again, this paper is available. It's really interesting, but these chimeras were bred with female bustard and essentially did lead to more of these eggs being created. So
yeah, that is very cool. I am going to say though, those weren't extinct.
No.
They weren't getting like revived. So it's like endangered species, but you do have the numbers there already. So it's
again, proof of concept.
okay.
So if you have a very few or an animal that dies, but you have like if you manage to create one and you could get cells from it, I don't know. It was just something I thought was kind of interesting. As this sort
it
of
definitely is.
De-extinction.
Oh, that's a nice term.
I think that's what it's called.
Okay.
When you're trying to like get animals back
Yeah.
from being extinct or from the brink of extinction.
Yeah, no, that's very cool.
Yeah. So thank you for listening to me talk about dinosaurs.
That's
and
okay. I
DNA.
enjoyed it. I feel like I learned a lot.
I've learned a lot
Oh.
from researching it. Like I thought I knew a lot about DNA as a geneticist, and obviously I know quite a lot, but learning about the genetics of birds and dinosaurs and how it degrades and how currently Jurassic Park is definitely remaining in science fiction. But I think it was cool, and I maybe one day, but maybe not, and maybe that's for the best.
Okay.
Don't
say gay.
Don't say gay.
No, I don't know. I was trying to segue into your thing.
Is this what it is? Yeah,
just tell us about how Margaret Thatcher ruined your life.
Is this going to be the crux of the episode? It's going to be like 6 hours long. Do you want to be more specific?
No. So, we've now reached the portion of the episode where we reflect on how it's been growing up LGBT, working as an LGBT person.
Yep.
We both work in academia, so that's quite a specific environment, I guess. Don't know if you had any thoughts, feelings, ideas, concerns, expectations.
Expectations. So let's not get into that. So yeah, okay. Do you want to talk about Margaret Thatcher?
Always. No.
So what Eleanor is alluding to, in case you don't know, in case you're too young to remember, or you're not from the UK, or you just don't remember and have blocked it from your brain, there used to be a thing in the UK, a law called Section 28. And you've probably heard about it if you've watched Drag Race UK or things like that.
That was the first time I heard about it.
Yeah, so it was a policy / law / guidance / I don't know what you call it, a mess, which was implemented by Margaret Thatcher and the Conservative government in the 80s. And it didn't go out of effect until 2003.
Yes.
And basically what this boiled down to is stopping, and please imagine the biggest quotation marks you can imagine, the promotion of homosexual lifestyles, ideals, I don't know what you want to call them, but essentially...
non-traditional families? Was that some of the terminology?
Something like that, yeah. But essentially what it did is it blocked teachers and people who are involved in the education of children and young people from being able to talk about being queer. Obviously, at the time, it mainly focused on homosexuals, but this spiraled out into anyone who was remotely queer, basically. And it covered the entirety of my schooling. And it was a weird one for me because as I grew up, my family are very supportive and very lucky. My mum has... a lot, and I was exposed to a lot of gay people growing up. We used to watch a lot of queer cinema. So like Priscilla, Queen of the Desert.
Oh, I love Priscilla, Queen of the Desert.
Rocky Horror, I guess, as well.
Yes, yeah.
Beautiful Thing. Have you ever seen that one? It's a British film.
Oh, it's not ringing a bell.
No, it's set on a council estate it's in South London.
Okay.
But it's about a young gay couple.
Okay.
And yeah, it's so basically I was exposed to a lot of this outside of school. Like my mum was very open, she had a lot of gay friends. I was very lucky to not have any sort of like negativity around that kind of thing. But going to school, it was not mentioned or talked about in any way, shape or form. And obviously at the time I didn't know what Section 28 was. No. It's only from looking back that you really realise how sort of like limiting that is.
Yeah.
For a young person. Again, I was very lucky, but a lot of people I know, friends of mine who are queer, never got to see that kind of thing, especially if they had slightly homophobic families or whatever. And so it took a long time for a lot of people, including myself, to come out because we weren't exposed to those sorts of things outside of the home. You know?
Yeah, and I suppose if you didn't have that sort of family life that you had, that's no exposure for you.
Yeah, exactly. Like I consider myself extremely lucky that even though at the time I didn't consider myself to be a queer person because, you know, we can come on to the history of bisexual erasure later, but you know, I knew gay people existed. I knew gay people. So therefore I'm like, this is a legitimate lifestyle and these people seem happy and normal. Therefore, this is all good. But, you know, and you're younger than me, so maybe this wasn't such a big deal. But, like, gay was still an insult. And,
yeah.
Yeah. Like, oh, you're gay, whatever.
That's so gay.
It's so gay. To me, that wasn't a term I aligned with being homosexual. It was just like, this is a bad word, even though it's the same word. You know? There's cognitive dissonance.
Yeah. And I feel like people... At the time, I don't feel like people sort of, it was just a thing you said, like, no, like you were a teenager, you're an idiot, and you didn't like think about it like too deeply. You just said it. But obviously the roots of it were like deeply sort of homophobic. Yeah. You know, I think especially when you're that age and you're young, you don't connect these things. But they can do harm. So when I say like, oh, you just didn't really think about the meaning behind it. Like it does do harm, and like the impact that it has is like damaging. But you just didn't really connect that because I mean you're a teenager, you're like a small psychopath.
Well, earlier than teenager as well.
Yeah,
like
you're just
school.
you don't have that sort of like knowledge of the world, I guess.
No.
to
And you
realise
have
what you're doing.
You have even less of it when you're a child under Section 28.
Yeah, exactly. Yeah.
Because you're already demonizing the
Yeah.
being gay, because you're not even allowed to talk about it. It's taboo. It's like witchcraft.
Yes, yeah.
So that was like Section 28 is obviously something that's affected a lot of young British queer people, especially of my and your age, and in between. So yeah, I think that it's definitely something we shouldn't forget, because it didn't make gay people go away.
No.
It just made lots of young people, myself included, have very complicated feelings about their queerness.
Yeah.
What else do we want to talk about?
How, with this sort of lack of queer representation in your sort of schoolhood life, but maybe not so much in your family life.
Yeah.
How did you, because like the default is that straight, and I feel like we didn't grow up in two dissimilar eras.
No. She lies to herself.
I'm so young. Joking. Please cut that out.
No. I'm editing this.
I feel like you're very much, and I hope it's not the same today. But like it was very much like the default is straight. You had to work out that you weren't straight, and you sort of, I don't know, like how did that happen for you?
I don't know if there was ever like one moment. I think that as someone who is basically bang in the middle of bisexuality, I'm equally attracted to all genders. I think that because I wasn't only attracted to girls and I did have attraction to boys, I just instantly heteronormatived myself into a corner and basically was like, well, I'm not gay, therefore I must be straight. And I think that a lot of that comes from the portrayal of bisexuality when I was young. Another piece of media which has traumatized me greatly is The Haunting, which is a movie that came out I think in like 1999.
Okay.
And if you've seen The Haunting of Hill House on Netflix,
yes,
from Mike Flanagan.
I've seen that one, but I think I also saw like the Haunting of Hill House.
It's just called The Haunting, but the 1999 movie.
Okay, maybe I saw a different film, but I saw A Haunting.
But essentially it's the same story, but done in a movie rather than a series.
Okay.
And one of the characters in it, who's played by Catherine Zeta-Jones, gorgeous, 1999 Catherine Zeta-Jones.
Always gorgeous.
Still gorgeous now, but especially 1999 Catherine Zeta-Jones. She's a bisexual character.
Okay.
And the line that is burned into my brain forever is, I have a girlfriend and a boyfriend and they don't like each other. Something to that effect.
Okay.
And so in my head, bisexuality, and this is common in a lot of media at the time, bisexuality was conflated with polyamory.
Yeah.
And I'm not polyamorous. And I am, so therefore I was like, well, I'm obviously not bisexual. Because,
clearly not.
Polyamory and bisexuality, and again, I didn't know the term polyamory at the time. I don't know if they even referred to her as bisexual, I can't remember. I think maybe they did. But I would basically was like, well, that's not me. So therefore I'm not bisexual. I'm just a spicy straight.
Spicy straight. Oh, God.
I just think girls are pretty,
Objectively
pretty. All behaviour has been disproved in terms of what came later. But obviously when I went to university, I experimented a little. I'd get drunk and kiss some girls, and I think that's one of the first times I learned about the male gaze. So that's fun, like making out with a girl in public really invites unwanted attention. But yeah, I fooled around with some girls, and again I was like, oh, it's just drunken, blah blah blah. And then by the time I was doing my PhD, so in my mid-20s, I'd kind of made the I'd realised that I probably wasn't straight. Especially because I spoke to more people who were like me.
Yeah.
And we chatted. And yeah, I was just like, oh, actually, no, this is fine. And this is a legitimate way of being. It's not conflated. Like all the bisexual people I met at uni and during my PhD were monogamous. And therefore I was like, oh, this actually is me.
Oh, okay.
And then I sort of like floated the idea to my partner at the time, and then it just became part of me. So I don't think there was one moment. I think there was like a realization of putting it all together, but I think it happened quite slowly.
Yeah. No, I see that. And I think now this is me talking about your experience, so please be like, no.
No.
But you mentioned like the aromantic label as well.
Yes, I did.
Is that something you sort of discovered later?
Oh, way later. Yeah.
Yeah.
I'm a terrible partner.
Oh.
And this is backed up by evidence. That's not true. I'm an okay partner. I feel like I'm a person who's very happy by themselves.
Yeah.
And again, in the sort of heteronormative straight thing, it's just assumed you're gonna like grow up and, you know, find a partner and maybe get married and maybe have kids and stuff. And obviously that's not the gay experience necessarily, but it is something that is just expected of you. And every single time I've been in a relationship, I've always felt that I never give my full self to it. Okay. I always hold something back, and I always feel, after a while at least, like once the honeymoon period's passed, once like the sort of, you know, sexual attraction is supposed to melt into romantic attraction, it just doesn't really happen for me. And so a lot of my relationships have ended with me just getting bored.
Okay.
A lot of relationships have ended with my partner literally turning around and telling me that I'm not a good partner, which again, fine, 100% fine. And the only relationship I would count as like successful was two, I guess. Like one was when I was like 16 to 18, when I was learning what relationships were. Obviously we didn't live together. Obviously we spent a lot of time apart. We went to different schools. So like we saw each other just to do fun stuff, and that was it.
That's cute.
And then the other relationship I had, which was very long, like 7 years long.
That is long.
It's a long relationship, but essentially we were just best friends who had sex.
Hey.
and like we lived together and stuff, but we had separate spaces and we had separate activities, and we used to go out separately as well as together. And I feel like that was not, I often feel, and I don't know if this is an aromantic experience, is that I feel very tethered in a relationship.
Okay.
Even though I'm not, even though all my partners have been pretty chill, not all of them, but a lot of them have been very chill and have allowed me to do whatever. But as soon as I'm asked to relent in terms of my time and my activities, I just find it really oppressive.
Okay. Yeah.
And I just don't find it that interesting. I think it's like I've never gelled with like rom-coms. I've never gelled with that kind of, like when people talk about their marriages and things, I just don't get it.
Yeah.
So yeah, that's something that came a lot later after a lot of some good, some not so good relationships. And I just realized this just not, I reflected on myself and said, I would be 100% happy if I never have another relationship.
Yeah.
Yeah, I think that it's just like the weight just left, you know?
Yeah.
I just felt so much more relaxed.
It is quite liberating, isn't it? When you sort of feel like identify these things within yourself that you didn't even know you were carrying.
Yeah.
And I think it's really interesting you saying about like rom-com films, because a lot of what you're saying about like being a bad partner, I feel like quite often you can be like sort of forced to therapize that away, you know, like, oh, you've got commitment issues and that's why. And like, I feel like it's good that we're sort of moving to a place where not everything is like an issue just because you don't have it within yourself to follow this one set path. Which I think is like most people really are breaking the mold somehow.
Yeah. I think that, you know, the internet, for all its flaws, has done some amazing things. And one of those things is seeing that you're not alone.
Yes.
So, you know, I had never met, not out at least, a bisexual person until I went to university.
Yeah.
So I didn't know that was an option. And I certainly hadn't met an aro ace person until I was in my 30s. Like again, people probably were, but they weren't out.
Yeah.
And so being able to see those things online and being able to see that this is a legitimate way of being and that you're not a weirdo for feeling these things, it's just really liberating. And it makes, and again, like meeting people like you and other colleagues we have who are queer, it just makes you feel much more comfortable to be yourself.
Yeah.
You know, you don't feel like you're hiding anything. Like you and I talk absolute bullshit about things all the time.
It's because I'm not very bright.
No, but you know, it's just fun to have that, because I've always like, again, been interested in queer things, but not really had a person 100% to explain those to until I was with my last partner. And it was very fun to be like, we're both queer people and blah, 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 blah. And it's fun to like make those jokes and stuff. And now I have that outside of a relationship. I have that as friendship. it's like just really nice, and it's really fun to make stupid bisexual jokes, and
Yes.
you know, getting that online as part of a community.
Yeah.
I know a lot of people are in places where they can't be out and
Yeah.
open.
Yeah.
So those spaces are life-saving.
Yeah. What was your sort of online?
Not good.
No, I mean, like, for me, it was Tumblr. And I was wondering...
Again, a bit old for Tumblr.
Yeah, changing space of the internet. Like, Tumblr is still going, but it's not the same as it was.
Yeah.
They banned the porn, and that's it. Died a death.
Yeah, well, that's the thing. I don't know what my online spaces are. I remember, again, before
Or
I was...
at the time when you were sort of exploring your identity.
Again, things like Pink News.
Pink News. Oh, okay. Yeah.
Again, I wasn't out to myself or anyone else, but I found it fascinating to read. And it became one of my bookmarks, like just to see what's going on in the world of queerness. Yeah, well, it was me trying to work out what the hell was going on with me.
Yeah.
What else was around at the time? I was mainly on like lolcats.
Oh, I remember lolcats. Yeah.
And even back then, like the internet was quite a hostile place to queer people, but you could always find people like you. I don't think there was like a sort of thing. I think it definitely came later once I was already out. Once I was like, and then I started looking for those communities.
Yeah.
And would spend time in them, reading them, not necessarily getting involved, but reading people's experiences and being like, hey, me too. Like, you know. I mean, I don't know how I didn't know sooner.
It is funny, isn't it?
It's so funny looking back and being like, oh, yeah, that makes sense. Like, I was absolutely obsessed with certain things that I probably would have been if I'd have been straight, but I certainly wouldn't have been as obsessed with them.
Example?
Musicals?
Go.
I don't know a lot of straight women like musicals. It's just...
Oh, yeah, let me tell you.
I was obsessed with them. Like Eurovision.
I was so into Chicago. I just like the jazz songs.
Yeah.
Not the sexy ladies.
Yeah, I think there's definitely a lot of that. I'm lucky I found myself eventually. I'm lucky I found myself even more now. I mean, we have spoken at length about bits and pieces to do with our experiences. But I think one of the things that I keep coming back to is when we did that panel about LGBTQ people. I don't know what that was for. What was that for?
Was it the Pride in Academia one?
Yes, I think it was that one. And like I kept coming back to the, you can't be what you can't see. Yeah. And that's why the internet is great. Like you can find someone who is like you. Yeah. Without very much effort. If you know the terms, or even if you don't know the terms. So yeah, I think that has really made things a lot better. Again, it hasn't made things perfect. There's still a lot of people who can't be themselves, and that is awful. I'm very lucky that I have a very supportive family and friends, and working in academia. It's not perfect, but I feel like it's more accepting than a lot of places. To sign this off, I guess, is that things are better than when I was a child. Certainly better than under Section 28. We still have a long way to go, as is the same with all progress. And I know that sometimes it feels like we are going backwards. Yes. But as anyone who's aware of British legislation at the moment, and what's going on in America and other places. But I personally am cautious but hopeful. And maybe that's just because of my small little bubble of academia. Like again, we have a tendency in academia to sort of like sit in our own little world and then be surprised when people don't align with us. Keep our eyes open. Help those we can. Like I know that it's sometimes hard to maintain momentum when things look bad. Yeah. But any small help that you can give to your community is appreciated. Yeah.
I quite like the phrase, I'm here, I'm queer, and I'm trying my best.
That is great.
Well, thanks for sharing, Bex. I feel like you've said welcome. a lot of quite vulnerable things, so I appreciate that.
I'm going to cry.
Don't cry because I won't know what to do. I have a packet of tissues in my bag, but please don't cry.
To be fair, when I'm crying, I don't want anyone to touch me, so that's fine.
Thank God.
This is why we're friends.
Yeah, thanks for sharing.
You're welcome.
I look forward to watching Jurassic Park with my nearly 6-year-old nephew and telling him, no, It's all incorrect. This is never going to happen. Shatter his little dreams.
I guess what I should say as well is that I love Jurassic Park. It's one of my favorite movies. And I know that it's just fun and it's science fiction, and I'm not trying to take away anyone's fun from it. I just think it's always interesting to underline science, like understand what is possible and what isn't.
I do. No, I completely agree. Obviously science fiction is very fun.
Yeah, it's great.
Sometimes people can get quite scared about things science related though. I was actually warned about this when I first started doing sort of outreach, just because I was like, yeah, let's talk about gross germs. And someone said, oh, like you've got to be careful not to scare children and people. So I guess, it's a lot of fun, but it is science fiction. And that's,
it would be unfun if there was no drama.
Yeah.
Like Jurassic Park wouldn't be as fun a movie if the dinosaurs didn't escape and eat everyone.
Yeah.
Like it would just be a nice day out at a theme park. And that's not
not a great film.
No, I mean, like it would be interesting. And apparently like the book goes into a lot more detail of the science and it's a lot more of the plot. Obviously I focused on the movie, but I think obviously I find that stuff interesting because I'm a geneticist, but obviously suspension of disbelief is so important for media generally. So
well, thank you for listening, and we'll hopefully catch you in the next one.
Thank you. Eleanor's going to be taking us through something cool next time, so
not scary.
Scary.
Yeah, scary.
Thank you so much for listening. See you next time.
Bye.